What is the name and cause of the autoimmune disorder known for swelling attacks of the face, throat, hands, abdomen, genital area, and feet? Hereditary angioedema. Do you know the autoantibodies that cause these diseases and syndromes? Occasionally, we come across these autoantibodies in the clinical lab in departments of serology, clinical hematology, clinical chemistry, urinalysis, and blood banking. Many of you are familiar with our topics in autoantibodies covered in several videos, and these are covered in many questions in the ACP study guide that tests your basic knowledge of these antibodies and links them to the specific diseases that they cause. Please preview two prerequisite videos: Immunology Basics, the complement system for the clinical lab, and autoimmunity concepts for the clinical lab. Hereditary angioedema is caused by a deficiency of a key complement regulatory protein called C1 esterase inhibitor. Coagulation and complement cascades are activated without proper negative feedback inhibition, with bradykinin causing leaky blood vessels to lead to swelling in the subcutaneous areas of the body, especially the face and hands. Fast forward if you've remembered highlights of the complement cascade. The complement cascade involves three pathways known as the classical, lectin, and alternative pathways. C1q is the recognition unit in the classical complement pathway, combining with subunits R and S to form the C1qRS recognition unit. Here we see C1 esterase inhibitor blocking esterification of C1s, thereby inhibiting formation of the C1qRS complex, an essential regulatory effect to provide negative feedback inhibition when the complement cascade becomes overactive. In fact, C1 esterase inhibitor is the only inhibitor of the complement cascade. The function of the C1 is to bind to the Fc portion of the antigen antibody complexes, while C1s and C1r are serine proteases to activate the cascade. The term complement fixation means that immune complexes complement the C1 cures at the Fc portion, and these antibody complexes are IgM, IgG1, IgG2, and IgG3. The membrane attack complex opens a pore that allows an influx of water and efflux of microbial electrolytes, causing hemolysis and microbial death. Note the regulatory effect of C1 esterase inhibitor to diminish formation of excessive anaphylatoxin C3A and C5A. Note that nomenclature of A after C3 and C5 represents anaphylatoxin activity. Clinical lab test methods include immunochromogenic, immunoturbidimetric, and nephelometry assays for individual complement measurements. CH50 titration assay is useful for total complement activity.